Hello everyone, welcome to Molecura. Today we're going to be talking about pyruvate kinase deficiency, its pathophysiology, symptoms, and treatment. So now we're going to be talking about pyruvate kinase deficiency, which is the second most common cause of hemolytic anemia worldwide. The first one being G6PD deficiency. So in our glycolysis, we said that we start with the glucose. This is our glucose, and we end our glycolysis with pyruvate. Now, there are a couple of reactions that happen, which is going to be important, and I'm going to be talking about it now. The first few reactions, I'm not going to write them in details since we already have a video on glycolysis. So there are a couple of reactions that happen to glucose, and we end up with a molecule that we call 1,3-bisphosphoglycerate, 1,3-BPG. Our 1,3-BPG undergoes a reaction, and it gets converted into a molecule that we called 3-phosphoglycerate. And the enzyme that was doing this was phosphoglycerate kinase. And in this step, one phosphate group was put on ADP to produce one ATP. So one ATP was getting produced in this reaction. Now, there is an alternative pathway which is called Rapoport-Lubring shunt. Rapoport-Lubring shunt. And this shunt is going to be particularly important in red blood cells. And 1,3-bisphosphoglycerate in this pathway can get converted into what we call 2,3-bisphosphoglycerate. And this is happening through the action of a mutase. So mutase enzymes, as we talked about, are moving the active groups on the carbon backbone. Here we have two phosphates on carbon number 1 and carbon number 3. And what's happening is that we are moving the one that is on carbon number 1 and we are putting it on carbon number 2. So this enzyme appropriately is going to be called bisphosphoglycerate mutase. It's called bisphosphoglycerate mutase. And this is particularly important in RBCs because 2,3-BPG can be used to decrease the affinity of oxygen to hemoglobin. And this decrease of affinity is going to cause better delivery of oxygen to the tissues. Now, this 2,3-bisphosphoglycerate can also get converted back into 3-phosphoglycerate, and this reaction also happens in this manner here. It's going to be converted back into 3-phosphoglycerate. And what is happening here, again, we want to know the name of the enzyme, that we have 2,3-bisphosphoglycerate. So there are two phosphate groups on carbon number 2 and carbon number 3. But here we have 3-phosphoglycerate. So one phosphate group is getting removed. And the word bis also is getting removed because now we have only one phosphate group. So we will lose this phosphate group here and we will lose the word bis, and we're going to have 3-phosphoglycerate. So the enzyme that is doing this is going to be called 2,3-bisphosphoglycerate. Let's write it here. It's going to be called 2,3-BPG phosphatase because it's removing a phosphate group. Now, these reactions are, as we said, important because our 2,3-BPG is going to allosterically bind to hemoglobin, and it's going to decrease the affinity of hemoglobin to oxygen. So this is going to be the importance of 2,3-BPG. That's why it's particularly important in red blood cells. So for the rest of the glycolysis, we're going to be having multiple reactions happening again, which we will not show all of them. And the last reaction that happens is going to be our phosphoenol pyruvate, which is getting converted into pyruvate. And this happens through the enzyme, which is the topic of our discussion today, and it's called pyruvate kinase. And through this, we are also important to note that we have 2 ADP getting converted, and there is a production of 2 ATP. Now, we have talked about fructose and galactose metabolism, and there were various types of pathologies associated with them. But when we talked about glycolysis, we are not talking much about pathologies. The reason being is that defects in glycolysis is incompatible with life. So most of the defects in the enzymes of glycolysis is going to be life impossible. So most of the individuals who are having a major defect in glycolysis end up dying in utero. But pyruvate kinase deficiency is usually not fatal because there are ways that the body can compensate for it, and we're going to be talking about it in more detail in a second. Another point to understand is that the RBCs are particularly affected by pyruvate kinase deficiency because they lack mitochondria. So they are dependent completely on glycolysis for production of ATP, because oxidative phosphorylation, such as electron transport chain and TCA cycle, is not happening in erythrocyte because all of those things happen in the mitochondria. So the deficiencies that we're going to have here is going to be what leads to the symptoms of pyruvate kinase deficiency. So the deficiency of this enzyme is going to be giving us our pyruvate kinase deficiency. Now, there is an important point, is that when this step is defective and it's not happening, these reactions are going to go backward, and we will end up shunting more 1,3-bisphosphoglycerate into 2,3-bisphosphoglycerate. So even though this condition is a hemolytic condition and the patients are undergoing hemolysis or having anemia, but they have a good exercise tolerance. Why? Because this is going back. This is going back and it's shunting our 1,3-bisphosphoglycerate to 2,3-bisphosphoglycerate, and this is decreasing the affinity of hemoglobin to oxygen. So oxygens are getting released from hemoglobin more easily, and they are getting delivered to the tissues. So tissues have a good supply of oxygen even though they are in an anemic condition. So this is the importance why we talked about this. Now let's go ahead and talk about the symptoms of pyruvate kinase deficiency and see if we can make sense out of it. So as we said, the main pathology that we are dealing with pyruvate kinase deficiency is going to be hemolysis of red blood cells. And how does this happen? So here we have a picture of our red blood cell, and our red blood cells are dependent on ATP for cation exchange. So we have something here that we call sodium potassium ATPase, and this is helping with exchange of cations through our RBC. So if you want to name it here, this is going to be our sodium potassium ATPase. And what happens that when the ATP levels in the RBC is depleted, we have depleted ATP level, this is going to inhibit this protein from functioning, from exchanging cations inside the red blood cell. So what we will end up with is with loss of potassium and loss of water. We're going to have loss of water, and we're going to have loss of potassium. So what happens is that our RBC gets dehydrated and gets shrunken, and this is what we call as echinocytes. So this is going to be our echinocyte. So when we do blood film, you're going to see a lot of echinocytes on the blood film because the RBC has got shrunken and has got dehydrated. So ultimately, this is going to lead us to RBC hemolysis. And this hemolysis is going to be releasing a lot of things in the blood which we can measure and we can diagnose this condition. And one of them, for example, being bilirubin, and one of them being our LDH, which we talked about in anaerobic respiration, which we'll come to again in a bit. So what happens here is going to be hemolysis, and we will end up with hemolytic anemia. So a patient comes with hemolysis and we would do a CBC, and what we can see on this blood count is going to be decreased hemoglobin. So on our CBC, we are going to see decreased hemoglobin. So we will have anemia, but we are going to see a normal MCV. So what we end up with is normocytic normochromic anemia. But as this goes on, we will have increased retic count, which is the reticulocytes getting released as a compensation. And increased retic counts can ultimately lead to an increase in MCV, and we will end up with megaloblastic anemia. So we will have increased MCV. Now, retic counts are going to be particularly important. Why? Because the reticulocytes have mitochondria in them. So our reticulocytes have mitochondria in them. They have mitochondria. So they can't do oxidative phosphorylation. And this means that they can bypass the deficiency that we have here, which is our pyruvate kinase deficiency. Why is that? Because this pyruvate ultimately would get converted into acetyl coenzyme A. And acetyl coenzyme A is going to go to the TCA cycle, and the TCA cycle will go to electron transport chain. But we have other sources of acetyl coenzyme A apart from pyruvate, and that could be beta oxidation of fatty acids. It could be catabolism of amino acids or ketone bodies. We talked about this on the video on overview of oxidative phosphorylation. Therefore, reticulocytes that have mitochondria can keep the show going. But the problem that arises is that when the reticulocytes get trapped into the spleen, which is an hypoxic environment, again, their oxidative phosphorylation will stop and they will end up going through the same problem again. That is why sometimes we can use splenectomy as a treatment for this condition. So splenectomy could be a treatment that we can offer to these patients. So the patients can undergo, as a treatment, can undergo splenectomy. And splenectomy is going to improve the condition because the reticulocytes are no longer trapped in an hypoxic environment. So splenectomy is going to improve the lifespan of reticulocytes. So another thing that we will see on the CBC is going to be a decrease in hematocrit. We'll have decreased hematocrit. And as we said, we're going to have things coming out of the RBC over here and get spilled into the blood. And therefore, these are going to be also good markers for a blood test. And what we will end up seeing is going to be LDH, which is lactate dehydrogenase. This is because the RBC is dependent on anaerobic respiration to keep the energy level and to keep glycolysis from going on. And therefore, we will have high lactate dehydrogenase in the RBC, and that's why we will have high lactate dehydrogenase in the blood when hemolysis happens. Another thing that we can see is going to be increase in indirect bilirubin. So indirect bilirubinemia is going to be a condition that we can detect in the blood as well. Sometimes children at a young age, they are going to be presenting with this problem. So all of this we can see on blood test. But what symptoms can we see? Symptoms are going to also be of that of anemia. So jaundice is going to be one of them. Let's try to write jaundice here. So jaundice is going to be one of them. So when there is a lot of hemolysis happening, the patient is going to become anemic. And the way that we are going to be treating this is with blood transfusion. And blood transfusion comes with its own problems. Sometimes repeated blood transfusion is going to end up giving you a state of iron overload. The same thing that happens with patients who are having hemoglobinopathies. Here we're going to have increased iron in the blood. An increase in the iron in the blood will end up needing iron chelation therapy. One other problem that we end up having is going to be gallstones. So patients due to chronic hemolysis will end up having gallstones, and usually during the first decade of life. So gallstones will end up needing a cholecystectomy. So most of these patients will end up needing a cholecystectomy to deal with this. So there are similar features when we have chronic hemolysis in a condition such as for thalassemia, for example. We have extramedullary hematopoiesis. So patients present, for example, with frontal bossing. So these things can also be seen in patients with pyruvate kinase deficiency. So an important clue is going to be the extramedullary hematopoiesis. So far, we have spoken about what pyruvate kinase deficiency is and what are the symptoms and what are the treatment options. Now, let's talk about the pyruvate kinase by itself as an enzyme. Pyruvate kinase usually has different types, and let's try to write them down here. So pyruvate kinase is going to have different isoenzymes. The first isoenzyme is going to be the one that is present in red blood cells, and this is going to be called PKR. Okay, so PKR is present in the RBC. There is another type which is usually present in the liver, and what we call it is as PKL. So pyruvate kinase deficiency usually are defect in these two enzymes. There are different other types as well. For example, we have the PKM. This is in a skeletal muscle. We have actually PKM1 and PKM2. So the defect is usually what we are dealing with is with these two enzymes that are our PKR and PKL. These are usually autosomal recessive, although there are autosomal dominant variants that have been described as well. So why is the liver not affected as much as the RBC, the same way that it's happening here? Because the deficiency of pyruvate kinase is not an absolute defect in the enzyme. It's just a reduced activity of the enzymes. Those who have this disease still are having around 15 to 20% of the enzyme activity. So the liver can keep things going. Furthermore, the liver is going to have some of PKM2 also present, which can help with this reaction going forward. For the RBCs, however, this is the only type that is present in the mature RBCs. But the immature red blood cells, such as the reticulocytes, have some residues of PKM2, which is going to help them still have this reaction going on. And as we said, the reticulocytes are going to have mitochondria, so they are going to have oxidative phosphorylation happening. So these are the different type of enzymes which can be deficient. There is a new treatment which is recently been approved by the FDA, we call it as a medication called Mitavpivat. Mitavpivat is going to be an allosteric activator of our pyruvate kinase. So this medicine is going to allosterically activate pyruvate kinase, and it's going to improve the activity of pyruvate kinase. As we said, this is not an absolute defect in the enzyme, rather it is decreased in activity. So by allosteric activation of the enzyme, we end up increasing the level of the enzyme activity. So let's try to recap. Our glucose in the glycolysis gets converted to pyruvate, and in the last step of glycolysis, we're going to have phosphoenol pyruvate getting converted to pyruvate through the action of an enzyme that we call pyruvate kinase. Deficiency of this enzyme could result in pyruvate kinase deficiency. There are 4 isoenzymes. We have the PKR, which is in the RBC, we have the PKL, which is in the liver, and the ones that are in skeletal muscle and multiple other tissues. Their defects are usually autosomal recessive, but there are types which are autosomal dominant as well. Symptoms of pyruvate kinase deficiency is going to be of hemolysis. So this is the second most common cause of hemolysis after G6PD deficiency. What happened is that our RBC is going to be dependent on pumps to keep the cation gradient in the cell. And these pumps are going to be largely dependent on ATP for functioning. So when we don't have pyruvate kinase deficiency, this step is going to be defective and we don't have production of ATP. Therefore, low ATP is going to make this enzyme stop working. What will happen is loss of water and loss of potassium is going to give us a shrunken and a dehydrated red blood cell that we call echinocytes, and this will result in hemolysis. As we have with hemolysis, the CBC is going to be showing us low hemoglobin with normal MCV. So this is a normochromic, normocytic anemia. We're going to have decreased hematocrit. Retic count is going to increase, and this can sometimes cause MCV to increase. As there is a lot of turnover, we usually recommend patients to take folic acid supplements, usually lifelong. Now, what happens is that reticulocytes usually have mitochondria, so they can undergo TCA cycle and oxidative phosphorylation, so they have a better chance of survival than mature RBC. However, when the reticulocytes get trapped inside the spleen, we're going to have an hypoxic environment, so their mitochondria will stop working, and this will again cause hemolysis. That's why splenectomy is going to be a good treatment. When the red blood cells are getting hemolyzed, we're going to have their content spill outside, and we can measure that as increased lactate dehydrogenase, and we are going to have indirect hyperbilirubinemia, which manifests as jaundice. Blood transfusion is going to be an important treatment as well, because there is recurrent chronic hemolysis, so blood transfusion sometimes can be important. But this is also something to keep in mind, that the repetitive blood transfusions can cause hemochromatosis and iron overload. Gallstones are a very common feature of this condition due to chronic hemolysis, and usually gets treated with cholecystectomy, and this can present in the first decade of life. Other features of chronic hemolysis and chronic anemias, such as extramedullary hematopoiesis, frontal bossing, and things that we see in hemoglobinopathies can also happen. So this is going to be our video on pyruvate kinase. Thank you for watching, and if you enjoyed this video, make sure to subscribe to our channel so you can explore similar videos on Molecura. Thank you.